Hello and welcome to BioCode. In this video, we will see how we can utilize ensemble database for variation analysis. And every species, most of the species on ensemble have variation datasets. For example, short sequence variants or long structural variants, such as disease or phenotype related variants information that can include deletion, single nucleotide polymorphism, or long structure variants or short nucleotide insertions as well. There are three types of variants available on NCBI to be precise: single nucleotide polymorphisms, uh, short nucleotide insertions or deletions. They can be insertions, they can be deletions, and longer variants that are basically classified as structural variants, and they are abbreviated as CNVs, which basically means copy number variations. So in this video, we will analyze human genome and some phenotypes and variations related to human genome. So I'm just going to search for I'm going to search for glaucoma, and we are going to analyze this phenotype that actually is a result of variation in the human genome. So we are going to analyze it. So as you see, I have searched within human genome glaucoma, and you can see that it is a phenotype, human phenotype. And just open the first or whatever you want to analyze, just open that page, and as soon until it loads, you will see a heading at the top that basically tells that it is a loci associated with glaucoma. Basically, the analysis that you will be doing right now is analysis on the gene or the loci or the marker that is available on the chromosome that has the variation as compared to other population. We are not actually analyzing glaucoma over here. We are analyzing the loci that is associated with glaucoma. HP tetra 0501 is actually the accession of this loci or this phenotype. But since we actually came from the search results, we don't need it now. So this is the table that actually provides the results. You can download this table by clicking on this link on this button, and that will actually download the file which will have a tabular representation of this dataset. In most cases, you don't need it, and it is obviously taking some time to actually retrieve the huge dataset. As you can see, this is a huge dataset. So I'm just going to get started with analyzing it. As you can see, the download has started. We will see at the end of this video how this data looks. Anyways, so all of these are, as you can see, RS basically means that they are variant accession numbers. All of them are variants that are reported in different genes at different regions within the genome of Homo sapiens, and they have some sort of variation within those regions. That is why they are corresponding to a certain phenotypic environment or uh, phenotypic lookout of the genotype that actually exists due to the variation. So if you see over here, you can see that a name is provided, type is provided, that it is a variant. You can also see structural variant. So what is the difference? Structural variant is basically, as we saw earlier, structural variants are longer variants. That means that there is a huge amount of variation provided. As you can see over here, that it starts from this point on the seventh chromosome and then goes to this point. So that means it is quite a huge variation, and it is actually not reported in a particular gene, but it is reported in a particular region. So, and it is also causing a phenotype disease, Ehlers-Danlos syndrome, which is a classic type, and this is annotated from DBWAR, which is an NCBI database for variations. So it also provides some sort of variant information wherever a single variation is present. As you can see, ninth chromosome, and the exact location is provided. And the plus sign basically means that this is a addition. This is a strand as compared to the negative, which basically means that it is a negative strand. That means reverse strand. So we'll find some negative strand. I hope we do. But some, as you can see, these are structural variants. Then the gene variant is also present. So there we go. We found the negative strand. So all of these are present, and you can just click any of them, and then we will be able to analyze them. Some do have the submitter information, external references provided. This is provided from the MIM, basically means OMIM, and that will take directly to a OMIM database, which is a human genotypic genotypic and phenotypic database. So what I'm going to do is I'm going to provide you an ability to put filters on your dataset. For example, let's suppose that you are looking for structural variants. So just turn all others and just turn on structural variant and just click apply, and then we will only see the structural variants and we will not see any other information because we don't need it. So in other cases we can also turn all of these other annotation sources. We can turn the syndrome, the phenotypes as well. For example, if you are looking for, since we wanted the glaucoma related disease, we do not need any other phenotype. So I'm just going to turn all of them off and just turn glaucoma on, and after that I'm just going to apply. Keep in mind that these filters change over time due to the fact that whatever search you are putting or whatever page you are on, these filters will change. So now we have structural variants that are related and that actually provide a trait or phenotype or disease, that is glaucoma. We do have annotation source, which is a database of variation, genotypic variation, and then we have external references that actually support this structural variant definition. So as you can see, we have genomic location of X chromosome, 11th chromosome second, Y as well second again, X again, and 17th chromosome. So all of these are corresponding to glaucoma. So let's just open one of them and then we will analyze that further. So this is the specific page for a structural variant, and since we actually came to this page from the search results, this was a loci associated with glaucoma. It represented all of the loci that was actually associated with glaucoma. Then we filtered out the structural variants and the definite glaucoma related disease related variants, and then we open one of them, and this is the page that we got. So at the top we have structural variant name, which is basically a weird name to be very honest, and then we have variation class. It is a copy number variation, so that means it is longer variants or structural variants, in other words. So if we click on this one, we will directly be taken to the sequence ontology database, which will define this variation in more elaboration. You can do that, but that is not the goal of this video, so I'm not going to do that. Then the most important information is provided that it is a gain in function as compared to some other variants. That could be a loss of function. This is a gain of function. A loss of function is demonstrated with the red color. So you can also find this information by going to this link, and this will directly be providing the information related to gain in this region, not the overall gain of the genome. Then we have the source database of genomic variants archive. Study was who did this study, that you can find over here. Alias is provided. This is basically the name, the secondary name of this copy number variation, which is basically the name on NCBI. I'm pretty sure if you search this alias on NCBI, you will be directly taken to this structural variant. Location is provided, forward stand. You can view this location in the location tab. I'm just going to open it in a separate tab. We will then see this exact location that is on the chromosome, and we will then see more information about this region, what genes are present, what accessions are present, that sort of information. So if you just click on variant table on this specific position, you will also find these variants that we are working on. We will also find these variants from this page as well. So let's suppose that you are on a specific chromosome and you are analyzing a chromosome and you would like to see what variants are present. You can just click on variant table and it will figure out all of those variants. It is obviously going to take some time because of the fact that this region has so many of the variants. So I'm just going to go ahead with the analysis that we were actually doing. So the secondary most important information is that, as you can see, it is actually providing us the exact genomic size of this variant, and this is a huge variant. As you can see, that this is why it is known as longer variant, so therefore as structural variant. Secondary information is that whatever information that actually corresponds within this region, what sort of transcripts are they corresponding to any phenotypes, is there any evidence available for that. So it is actually displaying us that there are 8 transcripts within this region. So if we just click on this particular region, we will then see all the genes that are present. I actually we can open this from this same page as well. I'm just doing it for the for the reason to reduce complexity of this analysis. So all of these genes are present within this region over here. Within this region, there are 8 transcripts. Within this structural variant, there are 8 transcripts that are actually different genes. All of them are, some are protein coding, some are long non-coding RNA structures, and obviously they would have some non-phenotypic effect on the genes. And then transcript coverage is provided, exons are provided, whatever exons are present within this region. Then we have more consequences of this particular gene. So that means there are 8 transcripts within this region, and all of them have copy number variations. You can see over here, and they have 5' UTR variant, intron variant. All of those variant information is present over here. You can just click on any of them, and you will be directly taken to particular that particular page of that particular gene. So I have opened now a gene from this particular hyperlink, and it will obviously take some time to load up. Anyways, under the table we do have more information, and these are basically providing information that whatever those variants actually caused that gene to change. For example, either is it a regularity region variant, or whatever region variant it is, it will provide more information on that. So all of them seem to be regulatory region variants, so I'm just going to display all of them. So all of them seem to be regulatory region variant. You can click one of them and you will be taken to that. So in the meantime we will see. Okay, so these are all the genes these are all the transcripts that are forming within this gene, and all of them seem to have some sort of variant information as well. So if you just go to genetic variant table, you will find variant information related to this transcript of Homo sapiens, and I think it is midline 1 gene, or gene synonymous are these are the names of this gene. So these are the variant table, and these are all of these variants that are available within this particular gene. We'll analyze this further. In the meantime, I'm just going to close this page as well. Let's open up the other page. Okay, so let's just analyze the other remaining part of this structural variant. Then we have an associated phenotype. So if we will just open this hyperlink, we'll then see glaucoma as the known phenotype. So now we have established that this structural variant is associated with one phenotype. We did not actually establish that. We actually saw it has to be a structural variant that actually causes a glaucoma. So it is pathogenic, as you can see, clinical significance. You can just make sure all of these significances from this hyperlink. And then we have ontology accessions as well, that will take you to other databases for more information. So what we have established from this analysis is that structural variants, how to find structural variants, how to find exact phenotypic relations between those structural variants, how we can find transcripts that are present within those structural variants, and how we can analyze them. So next we are going to analyze each of these one of these variants that are single nucleotide polymorphisms or short nucleotide insertions or deletions. We have already established the variants for structural variants, so I'm not going to go into that more detail. So I'm just going to open up the variant table, and the variant table has been opened for this particular gene that we found from these transcripts over here. So I'm just going to close this page as well, just to reduce the complexity. So as you can see, we are on the transcript page for this particular gene. It is present on this exact location on X chromosome on the reverse strand. This transcript has 11 exons and has 45 domains and features, almost 45, and it is also associated with 67,000, more than 67,000 variant alleles. So that means it is a hugely variant allele and is associated with many variant alleles, and it basically maps to 727 oligoprop. So if you just click on this link, you will see all of the entire list of these variant alleles. We will go into that detail later. So let's open up. We have this table. This table will provide more information about all of these variants that this gene is actually associated with. It is providing the variant ID, and clicking on this particular variant ID will open up more details about this particular variant as compared to the entire database. It is actually providing in a tabular information, and it is saying that A is changed with G and G is changed with A, that sort of information. We have a class which is single nucleotide polymorphism, as you can see, and it is sourced from DBSNP, which is basically database of single nucleotide polymorphism on NCBI, and it is a consequence or type of 3' UTR variant. We saw earlier most of them were 5' UTR, but this is a 3' UTR. We do have other information, but some of them is missing. So evidence is also provided. You can find the frequency, you can find more information about that. Then we have other information as well. For example, this, as you can see, this is a indel, short and nucleotide insertions or deletions. We have found that information as well. So what we can do, we can apply the same filters as earlier we did for different purpose, but we can apply same filters depending upon your needs. So I'm just going to turn all of them off, and I'm just going to find a more unique sort of variant for our gene. Let me just find, okay, exactly this one. So let's find the mature let's just filter class. Let's find deletion. There is only single deletion. Let's find this deletion. Turn all of them off, apply, and as soon as you apply, you will find a single deletion, and we have now filtered out all of the result, and we have this variant. So I'm just going to open up this variant for this particular gene, this particular transcript, and we will then analyze it in more detail. There we go. This is a variant information for a deletion. Same goes for the insertion as well, since it is a second type. SNP has the same sort of information as well. So most severe consequence of this variant is it is a 3' UTR variant. We can find all the other predicted consequences as well. The alleles it actually correspond is to either it can be a C or it can be a deletion. So ancestrally it was a C, or cysteine was present over here, or but over time it was deleted or whatever, something happened to it, and it is a deletion now. So change of tolerance score is provided, exact location is provided, BCF variants calling score is provided, and that sort of information is provided. So HGVS names are provided for this indel, exact indel, and DBSNP HGVS also provided. Ensemble and DBSNP have their own names, so be sure of that first of all. Then we have other information as well. For example, synonymous ClinGen allele registry is providing the more information about this. You can go to this link and you will be able to see more information on this. And source is basically it is a variant and indel, and it was found in DBSNP database release of 153. We can explore this variant in more detail. For example, we can have the genomic context, we can find the genes and regulation it is present in. we can find the flanking sequences that are flanking this region of deletion or insertion, whatever. We can find more frequency based information, for example population genetics. If some sort of disease or trait is associated with this exact deletion, then we can also find the phenotypic data, but it is not available. We can find 3D protein model if for some reason there is a 3D protein model available for some sort of contextual information, but it is not available. Same goes for sample genotypes and linkage disequilibriums. But would you have phylogenetic context? So I'm just going to open it up. So this is providing more information, genomic contextual information. There are 428 structural variants overlapping this exact region. So that means there are many overlapping structural variants in this region, and this red line is the exact region where it is actually participating for the deletion process. And you can find